Hey y'all, so today we'll be talking about phylogenetics. Phylogeny is the evolutionary history of a species or a group of species. A phylogeny for snakes and lizards, for example, indicated that scalyfoot, a legless lizard in Australia, originates from a different legged lizard than snakes did despite their phenotypic similarities. Their legless conditions evolved independent of one another. To construct a phylogeny, biologists use systematics, a discipline focused on classifying organisms and determining evolutionary relationships. Taxonomy is used to more specifically classify organisms. Taxonomy is the scientific discipline of how organisms are named and classified. Organisms are classified binomially, meaning their genus and species name are used to identify the species. Latin is used in the classification of these species, so there is a universal name for each species. The classification of each species is done in a hierarchical system beginning with the domain and ending in species. The taxon or taxa is the named taxonomic unit at each level of the hierarchy. For a leopard, Panthera is a taxon at the genus level, and if you look at the chart, it'll show you the entire hierarchy for the snow leopard. When constructing a phylogenetic tree, a branch diagram in which the evolutionary history of a group of organisms is displayed, it is important to remember that the tree represents a hypothesis about evolutionary relationships. These relationships are often depicted as branch points. The branch points are shown by the pink dots on the phylogenetic tree. Branch points show the divergence of two evolutionary lineages from a common ancestor. And then the sister taxa are taxons B and C, and they are two organisms that share an immediate common ancestor and are each other's closest relatives. Phylogenetics is studied because a species' phylogeny provides an enormous amount of information. For example, by analyzing the genes of corn or maize, researchers were able to identify two living relatives of maize that can be used to genetically modify the maize. In 2001, phylogenetics was used to identify the strain of anthrax bacterium as well that was sent to politicians and journalists so that authorities could track the anthrax back to the terrorist. Phylogeny systemists must first gather information regarding biochemistry, genes, and morphology. Biologists must determine if characteristics that two species share are due to homology or analogies. Homologies are similarities due to shared ancestry, whereas analogies are similarities due to convergent evolution. Analogous structures occur due to environmental pressures and natural selection, and the two species end up developing similar structures while evolving from two different species. The Australian mole and American mole look similar phenotypically, but when looking at their phylogenies, the Australian mole is really a marsupial whose young completes their embryonic development in a pouch outside of the mother's body. The American mole is a eutherian, and their young complete embryonic development in the uterus of the mother's body. The common ancestor of these two moles lived approximately 140 million years ago, and their lineages diverged about this time. The analogous characteristics evolved independently, and the two species developed characteristics as they became adapted to their similar lifestyles. Analogous structures that arose independently are also called homoplasies, which means to mold in the same way in Greek. Homologies are characteristics two species have in common due to shared ancestry. Molecular systematics is the discipline that uses DNA and other molecular data to determine evolutionary relationships. To determine whether two species have homologous or analogous structures, biologists must sequence DNA and compare the sequences of nucleic acids of organisms to determine the most likely cause of the similar characteristics. If two species are very closely related, the sequences differ at only a few sites. But if the species are distantly related, there will likely be different bases at many sites and may have differing sequence lengths because of insertions and deletions that have occurred over long periods of time. Two sequences that resemble each other at many points along their length are most likely homologous. Cladistics is the approach of systematics in which common ancestry is the primary criterion used to classify organisms. Biologists place species into groups or clades, which includes an ancestral species and all its descendants. Clades can be the same thing as taxa, only if the clade is monophyletic, meaning that the clade includes the ancestral species and all its descendants. Taxa and clades differ if the clade only includes the ancestral species and some but not all of its descendants, which is called the paraphyletic group, or by polyphyletic groups, which are groups that include taxa with different ancestors. Clades are defined by analyzing the difference between shared ancestral characters and shared derived characters. Shared ancestral characters are those that originated in an ancestor of a taxon. For example, all mammals have a backbone, but the backbone does not distinguish mammals from all vertebrates because all vertebrates have a backbone. The backbone predates the branching of the mammalian clade from other vertebrates. In contrast, shared derived characters are characteristics that are unique to a particular clade. In mammals, hair is considered a shared derived character because all mammals have hair, but hair is not found in their common ancestors. Phylogenies are developed using derived characters. The outgroup or a species or group of species from an evolutionary lineage that is known to have derived from the lineage of the in-group is the first species or group of species to diverge from the common ancestor. This group will not have the same derived characters as the in-group. The in-group is the species or group of species that we are studying. By comparing the members of the in-group, we can determine what characters are derived and then develop the phylogeny. Now, in creating a phylogenetic tree, branch length does matter. Branch lengths are proportional to the amount of evolutionary change or to the times at which particular events occurred. Even though the branches on a phylogenetic tree may be different lengths among organisms alive today, all the different lineages that descend from a common ancestor have survived the same number of years. The lengths of the branches differ because of the number of genetic changes that have occurred since the two lines diverged. There are two principles used to narrow the possibilities and create the most accurate phylogenetic trees possible. Maximum parsimony is the idea that one should investigate the simplest explanation that is consistent with the facts, whereas maximum likelihood is the idea that given certain rules about how DNA changes over time, a tree can be found that reflects the most likely sequence of evolutionary events. In maximum parsimony, you look at the facts you absolutely need in order to create the phylogenetic tree. With maximum likelihood, you look at all the facts and determine from the gathered information what is important for the phylogenetic tree. Phylogenetic bracketing is used to predict the features shared by two groups of closely related organisms are present in their common ancestor and all its descendants unless independent data indicates otherwise. The tree shows how the birds and Saurian dinosaurs are the most closely related of the species shown and how the two species share a common ancestor with the Orthician dinosaurs. Phylogenetics was used to split the original plant and animal kingdoms into what is now 3 domains and 6 kingdoms. The domains became eukarya, bacteria, and archaea as biologists began to realize the bacteria could be further classified. Kingdom Protista is separated from the animal kingdom recently for this reason too, and in the future will likely split into animal-like and plant-like protists. Horizontal gene transfer, which is a process in which genes are transferred from one genome to another through mechanisms such as exchange of transportable elements in plasmids, viral infection, and perhaps fusions of organisms, were all used to figure all this out. Thank you for listening.